Okay, well, thank you everyone for staying. With your permission, ladies and gentlemen, I'd like to change the title of my talk. So I've changed the title of my talk today to Seeking Perfection. My name is Michael Quail, team leader for sequencing research. Sequencing R&D at Sanger has three main functions: to evaluate new sequencing technologies and optimize them for production, to evaluate new equipment and molecular biology reagents, and to develop new protocols and applications for sequencing, some of which you'll see today. So this is the sequencing R&D group. There are some of us here at AGBT this week, including my boss, Harold Swerdlow, Manager of Development, Peter Ellis, Naomi Park, and Matt Mayhoe. Today I'm going to talk about Sanger and me and give some ideas of seeking perfection in terms of library kits, PCR enzymes, and finish up with some of the results we've got by the Archer from Enzymatics. So the spirit and the name of Sanger came from the Laboratory of Molecular Biology, Fred Sanger's group, and some of Fred's co-workers migrated the 9 miles south to set up the Sanger Center in the early 90s as a place to carry out pilot work for the Human Genome Project by sequencing the nematode worm C. elegans and the genomes of some important pathogens. And this is where I came in. I joined in 1997 to work on those pathogen genomes. And rather than sequence easy genomes like Craig Venter sensibly did, we decided to start on those that cause the highest disease burden, P. falciparum and Mycobacterium tuberculosis, which turned out to be very difficult genomes. Plasmodium falciparum causes malaria, responsible for 1 million deaths per annum, and has got a very high AT-rich genome. At the opposite end of the scale, tuberculosis, 1.5 million deaths per annum, but a very GC-rich genome with some areas in excess of 80%. So, having finished the human genome, the Sanger Center was rebranded as an institute, and today it's very much bigger. There are over 1,000 researchers within 40 different research groups aligned to our 5 main programs, and what underpins all of this work is large-scale genome biology, which is supported by our DNA pipelines, our R&D group, centralized sample management, genotype and microarray, capillary, 3 Illumina teams that take samples all the way through the sequencing process. We've got 30 HiSeqs, 5 MiSeqs, and a PacBio in production. And in my R&D group, we've got a MiSeq, 2 PGMs, and a proton. And we use all that sequencing power for looking at cancer genomes, studies in complex disease, metabolic disease, human evolution, regulation, pathogen genetics, model organism genetics, and stem cell characterization. So, seeking perfection. An example of the problem can immediately be seen from one of our initial data sets way back with Illumina, where we took 100x read depth. And in this genome browser, we plot read depth versus GC content. And we can see that in areas of low GC content, there are huge holes in the genome. And that's not perfect. And we needed to seek a solution to this. So we developed the PCR-free library prep and published that in Nature Methods in 2009. This worked very well, as I'll show you here. If we shred the malaria genome into Illumina-sized reads, plot their frequency versus GC content, you get a curve a bit like this. If you look at a library produced with PCR, you got a very GC biased curve, and a library without PCR, it was close to theoretical. And this worked very well, but without PCR, there's no amplification, and so it's only really applicable when you've got a reasonable amount of starting material. Problem is, your average patient with malaria is a child under the age of 5, and you can't get that much material. So a couple of years ago, I sought to find amplification methods that didn't cause bias. I looked at and published the results in Nature Methods. I screened over 40 different high-fidelity enzymes, different additives, different cycling conditions, made libraries and sequenced them for a set of 4 test genomes. The high GC genome of the whooping cough bacterium Bordetella pertussis, the neutral genome of the chicken pathogen Salmonella pylori, Staph aureus, and again malaria, because I believe that's the strongest test of any kit or sequencing technology. And we ranked the results by a low coverage index, and we defined this as the percentage of the genome that gives less than half mean coverage. So obviously, the lower the LCI, the low coverage index, the better. So we rank them in these graphs for each genome. And consistently, we find at the best enzyme end, there's one enzyme that's present in each case, which is kappa hi-fi. We developed these evenness of coverage plots where we plot genome fraction versus normalized read depth. And in these plots, we can instantly see how well a genome is covered, how evenly a genome is covered. Theoretically, you would get an even read depth coverage across the whole genome, but life isn't like that. Some areas are underrepresented, some areas are overrepresented, so you get a curve. And the closer that curve is to the red line, the better. So here we see for each genome, kappa hi-fi in purple, no PCR in green, and they more or less overlap each other. And fusion is a lot more biased in each case, particularly with malaria. With the GC-rich genome of Bordetella pertussis, you can see in this genome browser that in GC-rich areas, again, you get big dropouts in a very fragmented genome. So I followed that up in the present day, and we found another enzyme that's equally as good as kappa hi-fi, which is Takara prime star. Switching tack a little bit, I'm going to now talk a little bit about library prep kits. We've evaluated several different library prep kits, including the Spark kit from Enzymatics. I like the Spark kit, and I met somebody in the lobby yesterday who even described it as wonderful. It features ready-to-use lyophilized reagents. You don't have to thaw, you don't have to wait, which makes it very good for impatient or busy people like me. And there's no uncertainty. You know, sometimes you come to the lab and someone says, oh, I've left the freezer open, and you don't know what implication that's going to have to your experiment, whereas these just sit at room temperature and you just pick them up whenever you want and just use them. And we compared the Spark kit for each of our test genomes versus 3 of the major library kits that are available today, and it came out very favorably at a mapping rate that was very similar, gave very good complexities, and very low chimerism rates. We also looked to see if there's any GC bias against our GC-rich bacterium, Bordetella. It matched theoretical, and also pretty much matched theoretical with the AT-rich genome. So these are really remarkable results from enzymes that can just be kept on the bench and just used at will. So we've also used these enzymes for a real application for a process we call TRADIS, which is sequencing from transposon elements. Matt Mayo in the audience has done TRADIS from bacteria, and his colleague Irad has used the Spark kit for some mouse TRADIS libraries. What is TRADIS? Well, TRADIS is an application we published in 2009. It's sequencing from transposon insertions in the genome. So essentially, you shear DNA, genomic DNA containing a transposon, you ligate adapters, you enrich for transposon-containing fragments by PCR with 1 primer matching the adapter sequence and 1 primer matching the transposon sequence. And you sequence from those transposon insertions to capture the sequence of the genomic DNA where those transposons have inserted. And you can use this in bacteria to find essential genes. You challenge a bacterial population with transposon and transposase, such that on average, 1 cell contains 1 transposon insertion. You grow those up under control and test conditions, and you isolate DNA. You make a TRADIS library. You sequence them and map them against the genome, and you see some genes where you get virtually no reads, and these are the essential genes where that transposon can't insert because it will result in those cells just don't grow up. They're not viable. In mice, the flavor's a little different. We have mouse strains that express transposase, mouse strains that have a transposon. In their progeny, the transposon's free to jump around, and when it does, it inserts at random. In some cases, it'll inactivate a tumor suppressor, or it'll activate an oncogene, and you get progression to cancer. And we can sequence these mice that get cancer to see what gene has been mutated in order to give rise to cancer. So we've used the Spark kit quite successfully for this application. We've also looked at different streamlined library prep kits. Not the enzymatic one, I must add. These are ones that came out last year. These feature combined end repair and tailing steps, and quite often you can go directly onto ligation afterwards without cleanup. So overall, there are fewer cleanups, which makes the process more efficient. You get higher yield of fragment DNA at the end, and the whole process takes a shorter time. We looked at three different kits, which were differentiated slightly by their workflow. Kit A had a 65-degree A-tailing step. B and C were 70 and 72 degrees, I think. Kit A gave a higher yield, and we did, however, see a correlation in terms of bias with the A-tailing temperature. The 65-degree A-tailing temperature kit gave a slight increase in GC content in plasmodium from the theoretical 19 up to 21. And when you raise the temperature, just A-tailing temperature to 70 plus, we see a rise to above 23%. But even at 65%, you see a bias, this blue line here, in the plasmodium genome. So that's not quite as good as one might hope. Another issue we found with these kits, and we haven't had a chance to test the enzymatic reagents yet, is that when you compare the ligation efficiency of the Spark kit and the standard Illumina kit with that of these streamlined kits, you see a lower ligation efficiency. We can measure the percentage of fragments that have adapted to both ends by a qPCR assay, and this is much reduced in the streamlined kits, presumably because that A-tailing at the elevated temperature isn't as efficient. So overall, because of this increased ligation efficiency, although you get a higher fragment yield with the streamlined kits, you can sometimes get a lower library yield with these kits. Another thing that can go wrong in sequencing, another imperfection, if you like, is that sometimes you can get cross-contamination, sometimes you can get sample mix-ups, and when you do, that can be very costly and take a long time to resolve, and it can be embarrassing. So one thing we've developed, and we've just had published last week, was a process called SASSI-seq, sample assurance spiking sequencing. Very simply, this is a process where we generate a series of uniquely barcoded amplicons, and we spike those amplicons in unique combinations into samples as they come in the building. Those spike-ins at a low level, 0.1% or so, stick with the samples as they progress through library prep and sequencing, and we can look for their presence in the data sets afterwards, and we can make sure that each sample has the correct spike in. And that assures us that we've got the data from the correct sample. And when we see, this wasn't a mistake, by the way, we deliberately swapped this, but when you see a mix-up, you can immediately deconvolute it and see what's going on. So finally, I'd like to talk about gene targeting. This graph from our Tableau reporting tool shows the number of libraries that are produced by our institute for each month, color-coded by library type. Blue is adjuvant pull-down, so you can see normally somewhere between 25 and 33% of our libraries each month are targeted. We typically use SureSelect, which is good for larger target regions, but it's a long protocol. And the problem is we're moving to study smaller target regions in more and more samples, and it's potentially that's a problem. So what do we do? Well, Naomi in my group has been looking at evaluating targeted enrichment technologies. We've been looking at hybridization and PCR-based approaches, both commercial or homebrew. And we need to ask the question, do we need to invest in any capital equipment, like, for example, a rain dance machine or a fluid IM access array? So we've been looking at different bits of equipment and looked to see that in that target enrichment space below SureSelect, is there a one-size-fits-all solution? And ideally, we'd find the approach that gives the least bias. And as I say, finally, the approach that complements our projects and fits into that space beneath SureSelect. So what do we want? Well, we want both custom gene panels, and we want to be able to sequence ideally small numbers of genes in very large numbers of samples, because that's clearly the way things are going. So any target enrichment solution has got to be flexible. And in some extreme cases, we want to sequence single genes or parts of genes in tens of thousands of samples without the pain of doing individual PCRs and pooling them together. Any test has got to be fast and easy to perform with minimal hands-on time, and it's got to be reliable with good uniformity. And because quite often in these types of approaches, you can see bias. So here in this scatter plot, for a typical multiplexed targeting experiment with a well-known multiplex target enrichment solution, we plot read depth on a log scale versus GC content, and you can see there's considerable bias with over 3 logs of variation. And this leads to a requirement to sequence quite often to over 1,000-fold read depths, which is obviously very wasteful. So we've teamed up with one of our faculty members at Sanger, George Vasiliou, who is also an oncologist at the local hospital. His interest is in AML, and he wants to introduce a fast turnaround clinical assay into the clinic at the local hospital, Addenbrooke's, and we've teamed up to him to look at gene enrichment solutions for AML, both on all the 23 or so genes that are implicated in AML, and also by targeting individual genes. So this is where we've spoken to the people from Enzymatics, got an Archer panel against the DNMT3A gene. Why this gene? Because it looked to be the most difficult one. It had some regions of high GC content. It had some regions. It's got 23 exons of variable sizes. Quite often, when you've got some large exons with PCR targeting-based approaches, it's rather difficult to get through them because the amplicons overlap. So we took delivery of our first panel about a month ago. We ran this against 8 different clinical samples, and this shows the coverage across exon 18. And actually, we got probably what you might call quite variable coverage. But as pointed out earlier, you've quite often got to do rebalancing in these sorts of experiments. And once that pool was rebalanced, the coverage was very even indeed. And when we visualized this on a box plot for the original Archer experiment and the Archer experiment 2, so this is a normalized depth on a log2 scale, you can see that the coverage distribution across the entire gene has tightened really significantly. And where we compare it against 5 other competing major target enrichment technologies, you can see that it's very impressive and very tight indeed. And the uniformity in terms of the often quoted 0.1 and 0.2 of the median is up way above the 98% level and much better, as I say, than these other approaches. So that's it for today. I'd just like to conclude by saying that bias, like love, it is Valentine's Day, is all around us. And methods exist that are a lot less biased than others. And I believe that the use of the method of least bias should give more complete and meaningful data sets, which in the long term should save you time and money. Thank you very much.